Listeria is a genus of bacteria that contains 7 species: L. grayi, L. innocua, L. ivanovii, L. monocytogenes, L. seeligeri, L. murrayi and L. welshimeri. Named after the English pioneer of sterile surgery Joseph Lister, the genus received its current name in 1940. Listeria species are facultatively anaerobic, gram-positive bacilli. The major human pathogen in the Listeria genus is L. monocytogenes. It is usually the causative agent of the relatively rare bacterial disease, Listeriosis, a serious infection caused by eating food contaminated with the bacteria. The disease affects primarily pregnant women, newborns, adults with weakened immune systems, and the elderly. Listeriosis is a serious disease for humans. The overt form of the disease has a mortality rate of about 20%. The 2 main clinical manifestations are sepsis and meningitis. Meningitis is often complicated by encephalitis, a pathology that is unusual for bacterial infections. Listeria ivanovii is a pathogen of mammals, specifically ruminants, and has rarely caused listeriosis in humans. Background: the first documented case of listeria was in 1924. In the late 1920s, 2 researchers independently identified Listeria Monocytogenes from animal outbreaks. They proposed the genus Listeriosis in honor of surgeon and early antiseptic advocate Joseph Lister. However, that name was already in use for a slime mold and a protozoan. Eventually, the genus Listeria was proposed and accepted. All species within the Listeria genus are gram-positive, nonce-performing, catalase-positive rods. The genus Listeria was classified in the family Corynebacteriaceae through the 7th edition of Berger's Manual of Systematic Bacteriology. The 16 srRNA cataloging studies of Stackerbrandt, al. demonstrated that L. monocytogenes is a distinct taxon within the Lactobacillus bacillus branch of the bacterial phylogeny constructed by WAP paragraphs A. In 2004, the genus was placed in the newly created family Listeriaceae. The only other genus in the family is Brocothrix. The genus Listeria currently contains 10 species: L. fleischmannii, L. grayi, L. innocua, L. ivanovii, L. murrayi, L. monocytogenes, L. roquetii, L. seeligeri, L. stephanensis and L. welshimeri. Listeria denitrificans, previously thought to be part of the Listeria genus, was reclassified into the new genus Jonesia. Under the microscope, Listeria species appear as small, gram-positive rods, which are sometimes arranged in short chains. In direct smears, they may be cocoid, so they can be mistaken for streptococcus. Longer cells may resemble corynebacteria. Flagella are produced at room temperature, but not at 37 degrees Celsius. Hemolytic activity on blood agar has been used as a marker to distinguish L. monocytogenes among other Listeria species, but it is not an absolutely definitive criterion. Further biochemical characterization may be necessary to distinguish between the different species of Listeria. Listeria can be found in soil, which can lead to vegetable contamination. Animals can also be carriers. Listeria has been found in uncooked meats, uncooked vegetables, fruits such as cantaloupes, pasteurized or unpasteurized milk, foods made from milk, and processed foods. Pasteurization and sufficient cooking kill Listeria. However, contamination may occur after cooking and before packaging. For example, meat processing plants producing ready-to-eat foods, such as hot dogs and deli meats, must follow extensive sanitation policies and procedures to prevent Listeria contamination. Listeria monocytogenes is commonly found in soil, stream water, sewage, plants, and food. Listeria is responsible for listeriosis, a rare but potentially lethal food-borne infection. The case fatality rate for those with a severe form of infection may approach 25%. Although Listeria monocytogenes has low infectivity, it is hardy and can grow in temperatures from 4 degrees Celsius to 37 degrees Celsius. Listeriosis is a serious illness, and the disease may manifest as meningitis, or affect newborns due to its ability to penetrate the endothelial layer of the placenta. Pathogenesis. Listeria uses the cellular machinery to move around inside the host cell. It induces directed polymerization of actin by the actor transmembrane protein, thus pushing the bacterial cell around. Listeria Monocytogenes, for example, encodes virulence genes that are thermoregulated. The expression of virulence factor is optimal at 39 degrees Celsius, and is controlled by a transcriptional activator, PRFA, whose expression is thermoregulated by the PRFA thermoregulator UTR element. At low temperatures, the PRFA transcript is not translated due to structural elements near the ribosome binding site. As the bacteria infect the host, the temperature of the host melts the structure and allows translation initiation for the virulent genes. The majority of Listeria bacteria are targeted by the immune system before they are able to cause infection. Those that escape the immune system's initial response, however, spread through intracellular mechanisms and are, therefore, guarded against circulating immune factors. To invade, Listeria induces macrophage phagocytic uptake by displaying D-galactose in their teichoic acids that are then bound by the macrophage's polysaccharide receptors. Other important adsones are the internalins. Once phagocytosed, the bacterium is encapsulated by the host cell's acidic phygolysosome organelle. Listeria, however, escapes the phygolysosome by lysing the vacuole's entire membrane with secreted hemolysin, now characterized as the exotoxin listeriolysine O. The bacteria then replicate inside the host cell's cytoplasm. Listeria must then navigate to the cell's periphery to spread the infection to other cells. Outside the body, Listeria has flagella-driven motility, sometimes described as a tumbling motility. However, at 37 degrees Celsius, flagella cease to develop and the bacterium instead usurps the host cell's cytoskeleton to move. Listeria, inventively, polymerizes an actin tail or comet, from actin monomers in the host cytoplasm with the promotion of virulence factor actor. The comet forms in a polar manner and aids the bacteria's migration to the host cell's outer membrane. Gelsolin, an actin filament severing protein, localizes at the tail of Listeria and accelerates the bacterium's motility. Once at the cell surface, the actin-propelled Listeria pushes against the cell's membrane to form protrusions called phylopods or rockets. The protrusions are guided by the cell's leading edge to contact adjacent cells, which then engulf the Listeria rocket and the process is repeated, perpetuating the infection. Once phagocytosed, the bacterium is never again extracellular, it is an intracytoplasmic parasite like Shigella flexneri and Rickettsia. Epidemiology, the Center for Science in the Public Interest has published a list of foods that have sometimes caused outbreaks of Listeria: hot dogs, deli meats, pasteurized or unpasteurized milk, cheeses, raw and cooked poultry, raw meats, ice cream, raw vegetables, and raw and smoked fish. Cantaloupe has been implicated in an outbreak of listeriosis from a farm in Colorado, and the Australian company GMI Food Wholesalers were fined $236,000 for providing Listeria Monocytogenes contaminated chicken wraps to the airline Virgin Blue. Prevention. Preventing listeriosis as a food illness requires effective sanitation of food contact surfaces. Alcohol is an effective topical sanitizer against Listeria. Quaternary ammonium can be used in conjunction with alcohol as a food contact-safe sanitizer with increased duration of the sanitizing action. Refrigerated foods in the home should be kept below 4 degrees Celsius to discourage bacterial growth. Preventing listeriosis also can be done by carrying out an effective sanitation of food contact surfaces. Treatment. In non-invasive listeriosis, the bacteria will often remain within the digestive tract, causing mild symptoms lasting only a few days and requiring only supportive care. Muscle pain and fever in mild cases can be treated with over-the-counter pain relievers, and diarrhea and gastroenteritis can be treated with over-the-counter medications if needed. In invasive listeriosis, the bacteria has spread to the bloodstream and central nervous system. Treatment includes intravenous delivery of high-dose antimicrobials and inpatient hospital care. Duration of hospital care will vary depending on how widespread the infection is, but is usually no less than 2 weeks. Ampicillin, penicillin, or amoxicillin are often given for invasive listeriosis, and gentamicin is often added in patients with compromised immune systems. Trimethoprim sulfamethoxazole, vancomycin, and fluoroquinolones can be used in cases of allergy to penicillin. For treatment to be effective, the antibiotic must penetrate the host cell and bind to penicillin-binding protein 3. Cephalosporins are not effective for treatment of listeriosis. Prompt treatment of listeria infections in pregnancy is critical to prevent the bacteria from infecting the fetus, and antibiotics may be given to pregnant women even in non-invasive listeriosis. These oral therapies in less severe cases can include amoxicillin or erythromycin. In addition to antibiotic therapy, it often recommended that infected pregnant women receive ultrasounds to monitor the health of the fetus. Higher doses of antibiotics are sometimes given to pregnant women to ensure penetration of the umbilical cord and placenta. Asymptomatic patients who have been exposed to listeria are not recommended for treatment. It is recommended that these patients be informed of the signs and symptoms of the disease and to return for medical care if symptoms present. Research: Listeria is an opportunistic pathogen. It is most prevalent in the elderly, pregnant mothers, and AIDS patients. With improved healthcare leading to a growing elderly population and extended life expectancies for AIDS patients, physicians are more likely to encounter this otherwise rare infection. Better understanding the cell biology of Listeria infections, including relevant virulence factors, may lead to better treatments for listeriosis and other intracytoplasmic parasite infections. Researchers are now investigating the use of Listeria as a cancer vaccine, taking advantage of its ability to induce potent innate and adaptive immunity. See also, 2008 Canada Listeriosis outbreak, 2011 United States Listeriosis outbreak, list of foodborne illness outbreaks, notes. References: Aprashami S.H., Torby D., Berlusima T.J., Epstein P.S., Shah D.B. Bacterial adherence and viability on cutting board surfaces. Journal of Food Safety 14, 153 172 doi, 10.1111 per Joule 1745-4565.19940 B00591 Exa, Zaifa Liu, Changyi Yuan, Stephen B. Pruitt. Machine Learning Analysis of the Relationship between Changes in Immunological Parameters and Changes in Resistance to Listeria Monocytogenes, a New Approach for Risk Assessment and Systems Immunology. Toxical Sci 129, 157 73 doi, 10.1093 KFS201 Allerberger F. Listeria, Growth, Phenotypic Differentiation and Molecular Microbiology. FEMS Immunology and Medical Microbiology 35, 183 189 doi, 10.1016 per second 0928-8244, 02, 00447-9. PMIDA 12,648,835 Bales D.O., Wilkinson B.J. Osmoprotectants and cryoprotectants for Listeria Monocytogenes. Letters in Applied Microbiology 30, 23 27 doi, 10.1046 per Joule 1472-765 x 2000.00646 PMIDA 10,728,555 Bredolt S., Morkonen J., Kwajampo K., Alanko T., Olafsson U., Husmark U., Stjerberg A.M., Wertain G. Microbial Methods for Assessment of Cleaning and Disinfection of Food Processing Surfaces Cleaned in a Low-Pressure System. European Food Research and Technology 209, 145 152 doi, 10.1007 per second 002170050474 GMS, Schrift H. Comparative Evaluation of Adhesion and Biofilm Formation of Different Listeria Monocytogene Strains. International Journal of Food Microbiology 62, 103 111 doi, 10.1016 per second 0168-1605, 00, 00406-2 Chen Y.H., Jackson K.M., Kia FP, Schaffner DW Quantification and Variability Analysis of Bacterial Cross-Contamination Rates in Common Food Service Tasks. Journal of Food Protection 64, 72 Euro 80 Davidson CA, Griffith CJ, Peters AC, Fidding LM Evaluation of two methods for monitoring surface cleanliness: a plus or minus ATP bioluminescence and traditional hygiene swabbing. Luminescence 14, 33 Euro 38 doi, 10.10020214 1 less than 33 AID bio 5 14 greater than 3.0 CO. 2i. PMIDA 10,398,558 Food and Drug Administration 2005. Foodborne Pathogenic Microorganisms and Natural Toxins Handbook, The Abad Bug Book. Food and Drug Administration, College Park, MD. Accessed, March 1, 2006. Foschino R., Picozzi C., Savardi A., Bandini M., Feroldi P. Comparison of surface sampling methods and cleanability assessment of stainless steel surfaces subjected or not to shot peening. Journal of Food Engineering 60, 375 Euro 381 doi, 10.1016 per second 0260-8774, 03, 00060-8 Frank, J.F. 2001. Microbial Attachment to Food and Food Contact Surfaces. In Advances in Food and Nutrition Research, Volume 43 ed. Taylor, S. L. San Diego, C.A. Academic Press, Incorporated 320 Euro 370. Gassainview, Hughes D., Hansbrough P.M. Methods for the Isolation and Identification of Listeria spp and Listeria Monocytogenes, a Review. FEMS Microbiology Reviews 29, 851 Euro 875 doi, 10.1016/JFEMSRE. 2004.12.002. PMIDA 16,219,509 Gombas D.E., Chen Y., Clavero R.S., Scott B.N. Survey of Listeria Monocytogenes in Ready-to-Eat Foods. Journal of Food Protection 66, 559 Euro 569. PMIDA 12,696,677 Heek D.M., Summers E.B., Wong ACL Attachment of Listeria Monocytogenes and Salmonella typhimurium to stainless steel and bun and rubber surfaces in the presence of milk and individual milk components. Journal of Food Protection 56, 479 Euro 484 Kalmakoff M.L., Austin J.W., 1XD, Sanders G., Boehner G.S., Farber J.M. Adsorption, Attachment and biofilm formation among isolates of Listeria monocytogenes using model conduct ions. Journal of Applied Microbiology 91, 725 Euro 34 doi, 10.1046 per joule 1365-2672.2001.01419 Exa, Kusi Mainingram H.D., Riboldi G., Hazeledger W.C., Boomerara Survival of Foodborne Pathogens on Stainless Steel Surfaces and Cross-Contamination to Foods. International Journal of Food Microbiology 85, 227 Euro 236 doi, 10.1016 per second 0168-1605, 02, 00540-8. PMIDA 12,878,381 Lin C., Takuchi K., Zhang L., Dodgm C.B., Mayer J.D., Hall P.A., Doyle M.P. Cross-contamination between processing equipment and deli meats by Listeria monocytogenes. Journal of Food Protection 69, 559 Euro 569 Lowe J.C., Donaci W.A. Review of Listeria monocytogenes and Listeriosis. The Veterinary Journal 153, 9 Euro 29 doi, 10.1016 per second 1090-0233, 97, 80005-6 McNeil S., Walters D.M., Day A., Glaros A.G., Cobb C.M. Sonic and Mechanical Toothbrushes. Journal of Clinical Periodontology 25. 988 euro 993 doi, 10.1111 per joule 1600-051x 1998 TB02403X Maxi fate of bacteria exposed to washing and drying on stainless steel. Journal of Milk and Food Technology 38, 192 euro 194 McInnes C, Engel D, Martin W. Fembrai damage and removal of adherent bacteria after exposure to acoustic energy. Oral Microbiology and Immunology 8, 277 euro 282 doi, 10.1111 per joule 1399-302x 1993 TB00574X. PMIDA 7903443 McLaughlin J. The relationship between Listeria and Listeriosis. Food Control 7, 187 euro 193 doi, 10.1016 per second 0956-7135, 96, 00038-2 Montville R, Chen Y.H., Schaffner D.W. Glove barriers to bacterial cross-contamination between hands to food. Journal of Food Protection 64, 845 euro 849. PMIDA 11403136 Moore G., Griffith C., Fielding L.A. Comparison of traditional and recently developed methods for monitoring surface hygiene within the food industry, a laboratory study. Dairy, Food, and Environmental Sanitation 21, 478 euro 488 Moore G., Griffith C. Factors influencing recovery of microorganisms from surfaces by use of traditional hygiene swabbing. Dairy, Food, and Environmental Sanitation 22, 410 euro 421 Parinai M.R., Pitt W.G. Removal of oral biofilms by bubbles. Journal of American Dental Association 136, 1688 euro 1693. doi, 10.14219/JADA Archive. 2005.0112 Rokert J. Risk Factors for Listeriosis. Food Control 7, 195 euro 202 doi, 10.1016 per second 0956-7135, 96, 00035-7 Salo S, Lane A, Alanko T, Stjerberg A.M., Wertain G. Validation of the microbiological methods Hygiekult Dipsilder, contact plate, and swabbing in surface hygiene control, a Nordic collaborative study. Journal of AOAC International 83, 1357 euro 1365. PMIDA 11128138 Schluch WF Overview of Listeriosis. Food Control 7, 183 euro 186 doi, 10.1016 per second 0956-7135, 96, 00040-0 Seymour I.J., Burfoot D., Smith R.L., Cox L.A., Lockwood ultrasound decontamination of minimally processed fruits and vegetables. International Journal of Food Science and Technology 37, 547 euro 557 doi, 10.1046 per joule 1365-2621.2002.00613 Stanford C.M., Srikant R., Wu C.D. Efficacy of the sonic air toothbrush fluid dynamic action on removal of supragingival plaque. Journal of Clinical Dentistry 8, 10 euro 14 USDA FSIS. 2003. FSIS rule designed to reduce Listeria Monocytogenes in ready-to-eat meat and poultry products. United States Department of Agriculture Food Safety and Inspection Service, Washington, D.C. Accessed, March 1, 2006, Best K.L., Todd E.C.D., Riser E.T. Improved quantitative recovery of Listeria Monocytogenes from stainless steel surfaces using a one-ply composite tissue. Journal of Food Protection 67, 2212 euro 2217. PMIDA 15508632 White W., Carson W., Hambrauser methods for calculating the efficiency of bacterial surface sampling techniques. Journal of Hospital Infection 13, 33 euro 41 doi, 10.1016/0195-6701, 89, 90093-5. PMIDA 2564016 Wu Yuan CD, Anderson D. Ability of the sonic aerial electronic toothbrush to generate dynamic fluid activity that removes bacteria. The Journal of Clinical Dentistry 5, 89 euro 93 Tsor P, Tsor T, Doyle MP, Robino Jr., Meng J. Development of a model for evaluation of microbial cross-contamination in the kitchen. Journal of Food Protection 61, 960 euro 963. PMIDA 9713754, Zatola EA, Sasahara KC. Microbial biofilms in the food processing industry, a plus or minus, should they be a concern? International Journal of Food Microbiology 23, 125 euro 148 doi, 10.1016/0168-1605, 94, 90047-7. PMIDA 7848776. External links: Listeriosis at DMOZ, Listeria Genome at Patrick, funded by the National Institute of Allergy and Infectious Diseases.